I would like to speak to you today about my favorite organ, the placenta, as we have heard in the morphogenesis session this morning. And I would like to speak in particular about two aspects, the impact of the placenta on fetal development, and then I'm taking it one step further removed into the uterine impact on placenta and then fetal development. So our work really, what I'm going to talk about today, goes back to this paper in 2018, which was a Wellcome Trust-funded strategic award termed Deciphering the Mechanisms of Developmental Disorders. And what we did as part of this award was to screen a large number of embryonic lethal mouse mutants for embryonic but also placental dysmorphologies. And what we found as part of this is that actually the placentas are far more often affected than what was previously thought. So about 2/3 of all embryonic lethal mouse mutants also have placental defects. Bear that in mind in case you happen to work with mouse mutants that are embryonic lethal or subviable. But also even in this broad survey that wasn't really structured in any other way other than these are embryonic lethal or subviable mutations, placental defects were demonstrated to be very closely correlated with abnormal heart and brain development. And it is specifically this placenta-heart axis that we wanted to pursue further after I moved from Cambridge to Calgary. So is the placenta a cause of congenital heart defects or merely a bystander was really the big question. There are a couple of genes for which a placenta-heart axis had been proven insofar as the placenta is really causative of the congenital heart defect. And why is this such an important question? Well, congenital heart defects are the most frequent birth defect, affecting almost 1% of live births and many more that don't make it to term. And of course, research into congenital heart defects has mostly focused on the heart itself and has barely ever considered things as remote as the placenta. But perhaps most importantly, depending on what exactly you read, there's a large fraction of CHD cases that remain unexplained. And this is really the part where we think the placenta may fill some of that knowledge gap. So what did we do more recently? We went back to these 103 mouse mutant lines and selected specifically 3 that have joint placental and heart defects. This means fetal heart defects. And this resulted in the selection of these 3 genes that are shown on the far right. And it is really the first time that I embarked on such a large program of work that was only driven by phenotype and not by pursuing a function of a particular pathway or gene, etc. I've listed what these genes are below. You can see they are fairly unrelated in function. So this was really merely driven by placental and heart defects. And for dissecting causality, it is of course now instrumental that you make conditional mouse mutants. And for making embryo-specific mouse mutants, this was fairly straightforward because the SOX2 Cre driver is very well established, and it drives Cre expression only in the epiblast lineage, which gives rise to the entire embryo proper as well as to the endothelial cells of the placenta, but leaves all of the trophoblast lineage untouched. So thereby, when you have a floxed allele, you can make embryo-specific conditional knockouts that have a functional placenta. The converse situation is much more challenging because there is no established Cre driver that drives pan-trophoblast Cre expression very robustly, irrespective of what you may read. A couple have been established, but they don't really work all that wonderfully, or they also have expression in the embryo itself, and that is not something we wanted in this case. So we thought, how can we circumvent this problem? And we took advantage of this allele structure of these UCOM and COMP alleles that have been widely established that really start as a gene trap, and they have the functionality of starting from that knockout first TM1A allele that you can convert that back into a functional allele by flip. So what we did is we took that original SOX2 Cre vector from Annie McMahon's lab and swapped out Cre to flip and made a new transgenic line, SOX2 flip. And I'm happy to say these really work beautifully. They are very robust. They drive flip expression in the entire embryo proper, as well as in the epiblast-derived lineages, i.e. the endothelial cells that you can see there in green on the far right, as well as the yolk sac mesoderm, but not in the trophoblast or primitive endoderm lineage. So we applied both of these conditional knockout strategies as well as the constitutive knockout to all of these 3 mouse lines. And in this slide, you have about 4 years worth of work summarized. And very briefly, rushing a little bit over this, for SSR2, we found that this fetal heart defect was really entirely driven by the embryonic lineage, intrinsic to the embryo proper, but the exact opposite was observed for ATP11A, where we find, if I talk you through this in a bit more detail, I don't have a pointer. On the top, we have the constitutive knockout. You can see the embryo is very growth retarded and very edematous, often very indicative of a heart defect. And the trophoblast-specific knockout has exactly the same phenotype. When you look at the micro-CT images, you see the ventricular septal closure defects indicated by the red arrow, as well as the very severe myocardial thinning here in particular of the right ventricle, whereas the embryonic specific conditional knockout is actually fine, survives to birth, survives into adulthood. So ATP11A is not required in the embryo itself. It is only required in the placenta to make embryo development happen normally. And the last gene, SMG9, is also interesting. It actually has a mixed contribution by both the placenta and the embryo to that heart phenotype. But overall, you can imagine this really triggered our interest in this ATP11A gene, and we wanted to understand what goes wrong in these placentas that somehow affects fetal heart development. And what happens in these placentas is that the so-called labyrinth layer, where all the nutrient and oxygen exchange takes place, is not well developed. And specifically, they have a problem in the maturation of a very specific trophoblast subtype called syncytial trophoblast. We heard a little bit about that this morning. These are fusing cells that form very large syncytia, hence the name. And in the mouse, we have 2 of such layers. The syncytial trophoblast 1 layer, that is indicated in blue here, and then underlying the more greenish syncytial trophoblast 2 layer, which abuts the endothelial cells of the fetal blood vessels. So for all nutrients and oxygen that has to come from the maternal into the fetal circulation, everything has to pass these 3 layers, 2 syncytial trophoblast layers and 1 endothelial cell layer. And what we find in these ATP11A knockouts is that the SYNT1 layer, so the layer that faces the maternal blood circulation, is the most profoundly affected. And you can see that in the staining by the much weaker staining intensity, as well as the disorganization of the vascular structure. We also took advantage of the fact that we can have trophoblast stem cells where we can use CRISPR technology to knock out our genes of interest. And we applied this to ATP11A and other genes that were our favorites. And what you can see here is really that these mutant cells are incapable of fusing at the same rates as normal wild-type trophoblast stem cells do. This is a membrane marker staining, ZO1, and what you can see is that the cells are larger and the membrane becomes ruffled as if it's trying to break down but doesn't quite manage. And this is a phenotype that we observed also for the paradigm placenta heart axis gene, PPAR gamma, as well as remarkably for that SMG9 gene, where I said to you this has a mixed contribution to the fetal heart development by both the embryonic and the trophoblast lineage. So it seems to all converge on defects in SYNT1 formation that are somehow causatively involved in this placentally driven heart defect. And this is really what we try to summarize in this relatively trivial picture, and what we published now almost 2 years ago in Nature Communications, really the whole story of what I summarized here very briefly. So what have we done? We have really done, to my knowledge, the first unbiased assessment of a placental contribution to the etiology of developmental heart defects. And of these N of 3 lines, we have one, the ATP11A line, which has really an entirely placentally caused herd phenotype. The other one, SMG9, with a significant placental contribution to the herd phenotype. Of course, from an N of 3, we cannot say 1/3 of congenital heart defects are placentally caused, but I think it gives you an indication, and perhaps it's fair to say that the placenta may be the culprit in a sizable fraction of the many unresolved cases of CHD, and at least open our collective eyes to the impact of the placenta on fetal heart development. And for us, what is perhaps even more important is that we now have a very particular cell type that we are chasing that is causatively associated with fetal heart development. Perhaps it's not the only one of the placenta, but it's certainly in the set that we looked at, it was the most affected cell type. So what we're now trying to do is to actually select genes or screen genes for causing SINT1 defects and then see whether those are all those that have placentally induced heart defects. So now, as I said, rushing a little bit through this, I wanted to go a little bit beyond this, because what we observed while we were mating all of these mouse lines and had crosses to establish our constitutive knockouts is that we really struggle to get sufficient litters for the ATP11A strain. And this was not down to small litters, but it was actually down to the fact that some females just didn't become pregnant. ATP11A is not imprinted, we tested for that. So to really go a little bit further, we then set up control matings, wild type by wild type, as well as heterozygous female by regular wild type like 6 males. And what you can see here is that in the blue dots, some females carry litters just fine, more or less, and others really fail to become pregnant. They usually have blood-filled uteri with perhaps one resorption in it, but otherwise it's just a swollen uterus. So they really fail to establish early pregnancy. And that's what sparked our interest in the uterine morphology. And we established knockout females through placental rescue to look what might actually happen with these ATP11A females. And what you can see here is that the luminal epithelium of the uteri, shown on the left-hand panels, is really highly disorganized. By E-cadherin staining in the middle, we trace the signals to make it more obvious. You have this multi-layered structure of more rounded cells losing that beautiful columnar structure of the luminal epithelium. CXADR is a tight junction marker on top, so you really can visualize more beautifully how disrupted this morphology is. And on the bottom of that panel, FOXA2 is a very powerful transcription factor that is known to be restricted to the glandular epithelium, but you see now very profound and pronounced signals also in the luminal epithelium. You can also see the more rounded structure of those luminal epithelial cells compared to wild type. So we obviously also have some misspecification phenotype between the glandular and the luminal epithelium in these knockout females. The glands on the right are also not entirely normal, even though they are not multi-layered in structure. They have more or less completely lost that CXADR staining on the bottom, and they also have a much reduced expression of stem or progenitor markers such as SOX9 that is known to mark these cells in the glandular epithelium. So, but this is not where we stopped, because what we observed is in our placental RNA-seq data, even of the wild-type conceptuses, we found that the wild-type placentas that developed in ATP11A heterozygous mothers tended to cluster away from all the others. And you see this in these red dots on the left. So these are trophoblast-specific genes that we filtered for, so it doesn't have to do with the deciduar that contributes to the placenta and carries, of course, maternal genotype. It is actually only the fetal genes, so to speak, of the placenta that are still different depending on in which mother these placentas developed. And when you look at some key genes, such as PEC10, which is a syncytiotropoblast expressed gene, it is much reduced in placentas that develop in heterozygous females, even though that placenta and fetus itself is wild type. So that obviously means that the uterine genotype impacts trophoblast development, that in turn, I'm going to show you, also affects fetal development, even though much milder, but nevertheless it is there, that in some wild-type embryos that developed in these ATP11A heterozygous females, you still have ventricular septal closure defects, fairly mild, but they are there at E14.5, and in the more severe cases, myocardial thinning of the right ventricle. Sorry. So this obviously seems to indicate that there is, a knock-on effect from the uterus, and specifically from the uterine epithelial compartment, that affects the trophoblast portion of the placenta, mediated presumably through the maternal fraction of the placenta, the decidua, that then in turn impacts fetal heart development. So I think this is fairly striking, because you have many cases of congenital heart defects, and you sequence that baby left, right, and center, but the problem was actually only in the mother and wasn't even inherited, but the origin came from the uterus or perhaps from some other maternal component. So this is really the food for thought I wanted to tell you here, and I want to acknowledge the people who really contributed to this work specifically. It is Dr. Bethany Redford there in the middle on the top, who was a postdoctoral fellow in the lab since 2019 and has worked with all of these mouse mutants and the heart phenotyping. She is now a research associate in the lab. And Alexa Krala on the bottom there, she is a recently graduated master student who has looked at the uteri of the ATP11A deficient females. And finally, my political statement of the day, you can ponder while we take questions. We feel very strongly and really are personally affected by this threat that's coming our way. So thank you.
I apologize in advance. I am terrible at timekeeping and calendars. So, Myriam, I messed up the date to my wedding.
Oh dear. You're still married.
I have a problem. Tilo, actually. Fellow Canadian. Anyway, questions for Myriam? At the back.
Hi, thanks for your talk. I really liked it. I'm curious to know if you have looked beyond the heart. Is there any other place in the embryo that, you also see the effects when...
Yeah, there's obviously also a placenta-brain axis, and this also came out in our phenotyping screen. We have not looked at that further, but other people are. There's sort of this neuroplacentology field. The other organ that in our micro-CT imaging came out, but also we haven't considered any further, are kidneys, which is a bit surprising almost, but yeah, kidney development seems also to be affected, and that's not as well known at all. But yeah, so it does go beyond the heart, but the heart is the most obvious, I would say. And perhaps at that stage, the most, that will kill the embryo. The brain development at that stage will not.
So many contraceptive drugs work by affecting the... Oh.
Okay.
Many contraceptive drugs work by affecting the morphology of the uterine lining. I'm wondering what the effects of drugs in these mouse models are. So it's not a transgenic reason for the uterus lining being different, and if that still has an effect on placentation and then half defects.
We obviously haven't tried any of those drugs, but I'm sure they are not benign. So in that sense, I think it's a good reminder to consider those uterine effects on placentation and fetal development. I mean, even the progesterone receptor modulators, they are all membrane-bound things. So yeah, I think that is definitely something to look into much more carefully. We haven't applied them to mouse lines.
May I?
There's one in the back with the mic at the moment.
Oh, okay.
Very fascinating talk, and of course I like the last picture a lot. I'm wondering two things. First, can you kind of say when the heart defect first presents itself? Because when you looked, it was relatively late. Right ventricle defects would be associated with a second heart field. And so I'm wondering, can you nail down when the heart starts to go wrong? And second, in mind of the talks earlier this morning, do you think that we have a metabolism-driven effect, or do you think this has more to do with, I don't know, genetic, epigenetic, regulatory mechanisms?
Yeah, so to the first part, we had Henry Sukhov as our collaborator, as a heart specialist in mouse heart development, and had long Zoom calls with him over the pandemic, phenotyping these micro-CT images. And we have second heart field defects, but in the ATP11A mutants, which are exactly the ones that are placentally driven, the effect seems to be a little later and not second heart field linked. It is really more this myocardial thinning and VSD, and may really more point towards a proliferation defect of the myocardial cells as opposed to a strict second heart field defect. In terms of how it works, well, this is of course where we're all scratching our heads. Hemodynamics is always one frequently brought up possibility. We think, at least I think, that cannot be the sole answer, because there are mouse mutants that have a similarly disrupted placenta but not a heart defect. Because if by extrapolation hemodynamics is the answer, you would say all mouse mutants that have a placental problem in that labyrinth layer should also have a heart defect, but they do not. So we have recently put together, cunningly, I have that slide right next, a review where we have laid this out in the various interactions between the cell layer of that fetal-maternal interface. We don't know how it works. Is it a metabolite? Is it something that is transported from the fetal to the maternal, from the maternal to the fetal circulation that doesn't happen? Is it a trophoblast produced factor that doesn't get produced or gets produced too abundantly? So yes, I was also very intrigued by the metabolite talk and perhaps had that fructose 1,5-bisphosphate. It could be a microRNA, but bear in mind, I think it's often forgotten the anatomical structure. So anything where this defect happens still has to cross another 2 cell layers. So it is a bit of a conundrum. We cannot just take a TS cell supernatant and throw it onto a cardiac organoid, which is what many people do, and say, hey ho, this is how it works, because those 2 things never face each other in reality, at least not directly. Yeah, this is, I think, the point I want to make. Bear in mind the anatomical structure of the placenta when you try to address this.
I think for the sake of time, Kate, we'll let that continue at coffee break, just to try to get back on schedule. Thank you so much, Myriam, for a great talk. All right, I would like to welcome up my next speaker, Natalia Sanchez-Soriano from the University of Liverpool, who's going to tell us today about understanding neuronal vulnerability during aging. And do you have a pointer that's going to work? Okay, good, great.
I think it works. Okay, good. Great, so thank you very much for giving us the opportunity to present our work here today. So we are interested in understanding what drives neuronal atrophy during aging and how these changes that occur during aging makes neurons more vulnerable of developing neurodegenerative diseases. So in order to answer these kind of questions, we are studying the cell biology of aging, and we do this using the adult Drosophila brain as a model to study this. So in particular, we are working on a series of neurons, which you can see in this image. These are visual interneurons, which are located in the medulla region of the optic lobe of the fly brain. And you can see in this image the axons and the axonal terminals here in green, where synapses are located. So with time, during aging, this tissue undergoes remodeling, as you can see in this image. So this is an image of the same neurons in an aged brain. And if we look carefully, we find that axons becomes thinner, so always compared to the young brain. So axons becomes thinner, they develop swellings, and the axonal terminals, which has these beautiful hand-like shapes, they also swell and they break down. So these kind of morphological changes are typical from neurons that are becoming atrophic during aging in different organisms. So we also find at the subcellular level that there are several changes occurring in these neurons. And today I want to focus on 3 of these changes. So one change that we find is that the autophagic vesicles are different in each brain. So autophagy is a pathway, it's a degradation pathway, which is important in order to maintain the health of neurons, specifically in the synaptic terminals and the axonal terminals, where synapses are faulty or superfluous organelles and proteins that get encapsulated within autophagosons. These autophagosons then are transported along the axons, and it's in the cell body where degradation of these proteins is going to occur through fusion with acidified lysosomes, which also contains degradative enzymes. So if we look at the axonal terminals of these H brains, we find, I don't know, maybe we can put the light a little bit down, maybe it's easy to see. We find that there is an increase in the number of autophagic vesicles, and these autophagic vesicles are also enlarged. However, if we look at the cell bodies of these neurons, and remember this is where the process gets finalized, we find that there is less of these autophagic vesicles. And importantly, we find that the percentage of these vesicles which are acidified is significantly reduced. And this is suggesting that in these neurons, in all brains, autophagy is not working too efficiently. Another cellular component that is also affected in these aging neurons is the macrotubule cytoskeleton. We heard about the macrotubule cytoskeleton in some talks this morning, but in neurons, in axons, I just want to point to the fact that macrotubules are very particularly organized. So they're organized in these parallel arrays, and this is important to give a structure to the axons. It is important to determine the shape of these axons, and also, they are used as roads in order to mediate axonal transport. So, for example, autophagic vesicles move along these macrotubules. We find that in the H brains, the macrotubule bundles within the axons of these neurons decreases. So there are less macrotubules, as you can see here. But we also find the macrotubules lose the bundle organization, so they become disorganized. And the third process that we find affected in these neurons is oxidative stress. So we find that in these neurons we find an increase in reactive oxygen species. This is not too surprising because it's a typical hallmark from aging tissues. Okay, so we wanted to know whether these affected processes that we find in the H brains, whether they are actually mediating the transition of these axons into atrophic axons. And we did a series of experiments. I just saw in a summary here. So what we did in this slide was to generate artificial conditions during aging where we will even further reduce the efficiency of autophagy, we will increase the production of reactive oxygen species, and we also make the macrotubule network even weaker. And some of the manipulations are here. So for example, we knock down several regulators of autophagy. We fed flies with toxins that increases reactive oxygen species, and we also use mutations, for example, in glutathione synthase, and we also reduce the level of different macrotubule regulators that we know they are important for macrotubules. And in all these conditions, what you see here is that the basal level of neuronal atrophy, which we are measuring here by quantifying axonal swellings, increases. We did the converse experiment where we now protected these processes. For example, we enhance autophagy, we decrease reactive oxygen species, and we protected microtubules during aging. So, for example, we fed flies with rapamycin, we use knockdowns of KEAP1, which is a repressor of NRF2, therefore we have more NRF2 signaling occurring, and we overexpress microtubule regulators. And under these conditions, we suppress the appearance of these axonal swellings, suggesting that these neurons are becoming less atrophy during aging. So we are proposing that all these processes are driving neuronal aging, and we are proposing that there is a cascade, so they are interconnected. So we propose that defective autophagy leads to an increase in reactive oxygen species. This is affected the regulation of microtubule networks, and it's this effect on microtubules which is triggering axonal and neuronal atrophy. So this is work in progress, but we have several lines of research which are really supporting of this aging cascade. And I just want to briefly show one of them. So this is a series of experiments that now we are doing using primary neuronal cultures from Drosophila. And what you can see here in these neurons, so these are the axons, and you see very bundled microtubules. You cannot distinguish microtubules with this kind of microscopy. But if we single microtubule, sorry. But if we now expose these or use conditions that will lead to increase in reactive oxygen species, for example, we provide paraquat or DEM in the media, or we use mutations in superoxide dismutases, then what we observe is that the nicely bundle of microtubules is affected. So microtubules becomes unbundled, and actually they become less stable. And this is what we are getting in our in vivo aging model. We can suppress this with Trolox, and this is confirmed this has to do with oxidative stress. Importantly, we also find that similarly, if we decrease autophagy, for example, by using vafolimycin or chloroquine, which inhibits the fusion of the lysosomes to autophagosome, or if we use mutations in regulators of autophagy, such as ATG1 or ATG8, we also increase these microtubule phenotypes, as what you can see here. But importantly, we are able to suppress these phenotypes if we now provide Trolox, which is the antioxidant. And this is suggesting that the phenotypes of microtubules, which are induced by defective autophagy, are related to oxidative stress, supporting this cascade. So we think that the microtubule cytoskeleton are key players in this cascade. They are downstream of autophagy and oxidative stress. But because microtubules are so important, for example, for autophagy, we think that they also initiate a feedback loop, making this process even worse. So this highlights the potential of using microtubules as a therapeutical strategy to improve aging. And our experiment shows that if we protect microtubules, then atrophy does not occur or occurs to a lesser degree. But this is also having consequences, if we think, in conditions of neurodegenerative diseases. So, for example, conditions, several neurodegenerative diseases are linked to increases in oxidative stress. And here I'm showing an example where flies are exposed to paraquat. Paraquat is a pesticide that has been linked to Parkinson's disease. And that these conditions of paraquat exposure, neurons becomes atrophic, but we are able to rescue this by overexpressing EB1, which is a microtubule regulator. And similarly, if we now have conditions where autophagy is decreased, and again, many different neurodegenerative diseases has been linked to defects in autophagy and lysosomes, if we use chloroquine in these flies, we induce neuronal atrophy, but again, we are able to rescue these phenotypes by overexpressing EB1, which is a microtubule regulator. So with this positive note on microtubules and how this can be used for therapies, I would like to finish by thanking the team. So my current team and past members of the team, this has been a really or it is, because we are still working on this project. It's a really teamwork, and without them, we would not be where we are. And of course, our funders, collaborators, many colleagues for sending reagents, and for you for listening today. Thank you.
Thank you. Hi, Natalia. Great talk. So I have a question about the other cytoskeleton components during aging. Have you seen any alteration of them?
Yeah. So I guess you're maybe interested in actin.
Or intermediate filaments, or?
Yeah. So Drosophila doesn't have intermediate filaments, but of course actin is very prominent, and we're just starting to look at actin. So we don't know yet, but yeah, it could be highly involved as well.
Okay, well, let's thank Natalia again one more time, and
so for the other questions, I will be in poster 71, so you can come and ask me directly.
Great, and I'd like to welcome up our next speaker, who's Loydie Jerome-Majewska from McGill University on the contribution of abnormal facial defects in SF3B4-related syndromes.
Great. Thank you very much. First, I'd like to thank the organizers for inviting me. It's been really lovely to be here in Liverpool. And let's see, how does that work? Okay. So today I'm going to cover one of the genes that we study in my lab, but really my lab in general is interested in understanding congenital malformations. And so what we do in the lab is we use the mouse to try and model some of the phenotypes that we see in human, and then try during developmental time to figure out when things go wrong. And someday, I hope we'll be able to try and change the conditions so that we can either rescue or prevent some of these abnormalities. But today what I'm going to tell you a lot about is how we make abnormalities. And so 3 of the syndromes that we're studying right now in the lab are cerebrocostomandibular syndrome, which are due to mutations in a gene called SNRB, Nager and Rodriguez syndrome, which I'm going to talk about some more, which are due to mutations in a gene called SF3B4, and mandibulofacial dysostosis with microcephaly, which are due to pathogenic variants in a gene called EFTUD2. And what these 3 syndromes have in common is that they are due to pathogenic variants in genes that are important for splicing. And splicing occurs in every eukaryotic cell. It's something that you might call housekeeping function. And yet, when you have these pathogenic variants, you get very specific craniofacial defects. And just to take you back, so the way that splicing works is that there are splicing factors that are recruited, and these splicing factors recruit the U1, which binds to the 5' splice site, U2, which binds to branch points, and then the 3' splice site is identified, and together a tri-SNRP complex consisting of U4, U6, U5 is recruited, and this complex, will initiate a catalytic reaction that leads to intronic sequence removal, and then ligation of the 2 coding sequences, or the exons together, to make a final mature mRNA. And so you might imagine then, we were very surprised when exome sequencing revealed that there were these pathogenic variants in SF3B4, EFTUD2, and SNRB. And so we've decided to focus on these 3 genes because they have these shared craniofacial defects that we're trying to understand. But I just want to point out that there are other members of the small nuclear RNA protein families that are important in splicing that also gives other craniofacial defects. So to get into the meat of today's talk, which is about those pathogenic variants in SF3B4. And so SF3B4 related syndromes, which is how we refer to Nager and Rodriguez syndrome in the lab, is associated with mandibular hypoplasia, preaxial limb defects, which you can see here. But there are also less common defects, such as trigonocephaly, cervical rib, and cervical hemivertebrae. And so the SF3B complex itself is quite important in the first step of recognizing that 3' splice site. And so SF3B4 contains 2 RNA binding domains that helps to attach the U2 to the 3' branch point site.
SF3B1 and SF3B3 are actually the scaffold around which the mRNA is attached. And then SF3B6 binds to the branch point nucleotide. And so this being a core component of the U2 is surprisingly mutated or has pathogenic variants in quite a large number of people. And in fact, as other members of this family, including SF3B2, has also more recently been shown to contain pathogenic variants in a subset of congenital malformations. And so this was identified in the early 2017, I believe. My God, time is flying. And so we decided to use CRISPR-Cas9 at that time to generate a mouse model. Coming from a mouse developmental biology lab, we are acutely aware that often in mice, when you delete essential genes or you make heterozygous or homozygous mutations, those embryos die during embryonic development and may not model your disease. And so what we did was we used CRISPR-Cas9 to generate these conditional mutant lines with LOXP sequences flanking exon 2 and 3 of SF3B4 with exon 2 and 3 encoding for those RNA binding domain. And what we showed was that if you delete this one allele and you generate heterozygous mice, those heterozygous mice have reduced levels of the mRNA and the protein. And Shruti Kumar, who is the PhD student who did this work, was very disappointed because these mice survived. However, they did have cervical, thoracic, and lumbar and sacral defects. We showed that this was due to reduced HOX expression and altered HOX patterning. And we found that there was increased missplicing of key chromatin modifiers, and we think that that's why they had these cervical defects. But we were interested in the craniofacial region, and so what we decided to do was to remove SF3B4 in neurocrest cells. To do this, we used Wnt1-CRE to delete SF3B4 in these neurocrest cells as they are being born. And so this is just a reporter line showing you that the CRE is expressed very early on and continues to be, the derivative of those CRE expressing cells will contribute to the craniofacial region. We were very happy to see that when we did this, we do get embryos that have malformations starting very early on in development at embryonic day 8.5, and then when we look at embryonic day 17.5, they have craniofacial defects that range similar to patients from very severe to quite mild with just a cleft palate. We did have one mouse that survived, so it's like 1 out of 1,000, but, and it was perfectly normal to this day. I don't know why, and I cannot answer why that was. What we showed was that in most of these embryos, if you look at neurocrest cell migration, some of them will have some neurocrest cells that have migrated in. This is the first arch that this arrow is pointing to, so this is the first branchial arch, while others will have no neurocrest cells. And I'm not showing you the data, but we also showed that at the time that these neurocrest cells are being born, there's increased apoptosis in a subset of these mutants. We went on to show that these cells are not just dying as they're being born, but also as they migrate through the mesoderm to get to the first arch, you see increased apoptosis. And so this is a tunnel staining showing you the red tunnel positive cells, which are also positive for the neurocrest cell marker, in this case, using the MTMG line. And then we showed that it's not just apoptosis that explains the cell death or the loss of these neurocrest cells. When they do get to the first pharyngeal arch, and so this is phosphohistone H3 staining, again, the neurocrest cells are in green. What you can see is that in the mutants, there are less green cells, so there are less neurocrest cell positive cells here. And then if you look at phosphohistone H3, there's reduced proliferation, and this reduction in proliferation is really in these neurocrest cells. And so what this told us is that these cells were being generated in the subset of mutants, and even when they were able to get out of the neural tube area and migrate into the first arch, they stop proliferating and can't form the structures that they should. And so what we did next was we took the heads of these embryos prior to phenotypes and then did RNA sequencing to try and find those splicing targets that are misspliced that explains this phenotype. This is not surprising to us, although it may seem surprising, was that the p53 pathway was very highly upregulated in these SF3 neuroCrest cell mutants. And so this is just showing you that if you do a branch point, go analysis, you see that the p53 pathway is increased. The reason I said that this is not surprising is that we had also found this to be the case in the EF2D2 and SNRB mutant. So the next question we asked was if this pathway is upregulated and maybe explaining the change in cell survival, what happens if we remove p53? And what we saw was that similar to EF2D2, when we removed p53, it did not improve survival or the phenotype. And so these embryos continued to be abnormal and failed to survive and to have craniofacial defects that looks exactly like p53 wild-type controls. So then we turned our attention to splicing. And here we started seeing some interesting things. Unlike the other mutants, there's a huge increase in genes that have skipped exons. And so what that means is that during the splicing reaction, you can have an mRNA that has these 3 exons, for example, the purple, green, and orange. And in some cases, you can have a process called alternative splicing, where the green is not included, and so you have a transcript that only has this purple and the orange. And so what this means is that in these mutants, we have more of these cases where you have the splicing and there's an exon that's missing. And we did find in the RNA sequencing that some of the genes that we know that are important for neuroCrest cells are included in this group of genes. And right now, Josh Moore in the lab is really trying to dig into the splicing using single-cell sequencing. What was interesting was the biology that we started getting information on. And so when we looked at the group of genes that were differentially spliced and that had these skipped exon events, what we saw was that the length of the genes that had these events in SF3B4 were smaller. And then when we looked at the skipped exons itself, you can see here that they were also smaller. And then when we looked at the upstream intron length, they were smaller. So what this meant is that there's a very specific subgroup of genes that are impacted by the reduced levels of SF3B4. And so we asked the next question, which is, well, is there some kind of a sequence that we could find that could identify these genes? And this was one of those emails you open and you said, okay, this can't be true, but it was, after we looked at it multiple times, was that the distance to the branch point was reduced, but there was this sequence that was showing up in those genes that are showing these alternative skipping events in SF3B4. And so I was just telling someone, so now we have set up a collaboration with someone who actually studies the mechanism of splicing because we want to dig into this further to understand what this means. But all put together, what we found was that the exons that had events, when we look both at the heterozygous as well as these neuroCrest cell mutants, tend to be skipped events are in genes or are in exons that are AT-rich and GC-poor, and so that's just what this is showing you here. And when you look at the gene composition altogether, you don't see this change. And so it's really those exons that are being altered by SF3B4 that have this landmark. And so what we have now identified is that SF3B4 is necessary for efficient splicing of genes that are smaller and that have these very specific sequences. And we think that this means that if we look at neuroCrest cell regulators, those subset of genes that matches what we think is important for SF3B4 will be the ones that are most impacted. And so coming back to all of these mutants, so you make all of these mutants, they have similar craniofacial defects, the patients have similar craniofacial defects. Can we identify something that explains why these craniofacial splicomopathies exist? And so when we look at our data, what we find is that mutations in SNRB, EF2D2, or SF3B4 all result in increased p53 activity because of missplicing of this key p53 regulator MDM2. However, the phenotype is only improved in SNRB mutants when you remove p53, suggesting that other things are involved in this phenotype in EF2D2 and SF3B4 mutants. And so what we have then is that we have increased exon skipping in all of these splicomyopathy mutants. And it's only a handful of these splicing events that are shared. And so now we're trying using HCR and other methods like long-read single-cell sequencing to understand what these genes are and what they have in common, if anything. But there are differences between these different genes because, for example, when we look at SNRB or EF2D2, we haven't identified any differences in the signal that surround those exons that have skipped events. And so we're really excited to study this some more because what our data is suggesting is that these splicing genes are important at all stages of neurocrest cell differentiation, and that survival is an important component of the phenotypes that we see. I haven't talked about this, but we're also exploring changes in the epigenetic landscape because it's not just in the vertebrae that we found that these things are differentially spliced. We're also finding differential splicing of epigenetic modulators in the neurocrest cells. And so Yan Chen is currently looking at that in the lab in more details. And we're really excited to set up our collaboration with Karine to really look at what's going on at the splicing, at the level of splicing and how splicing is being regulated using these mutant mouse lines. So I want to thank you. I am right on time. And I'd like to thank Shruti Kumar, who was the PhD student that did all of this work. And then Yan Chen, Janelle, Josh, and Marie-Claude are continuing on the splicing mutants, and then Amanda and Cassandra are looking at TIMED2 and protein transport, which is the other half of the lab. Thank you again for the invitation.
There's one at the back up here. Maybe I'll start with one question as we're running the mic. So I had a question about how differential splicing in different tissues is going to be affected, and whether you're going to see clustering of hits on chromatin modulators, things that might be dosage sensitive transcription factors and everything else under these patterns that you're starting to look for.
Yeah, so we've used neurocrest cell mutants. We've also made MESP mutants and now T-CRE mutants. So we're using a number of different CRE lines. And what I can tell you is that at least for SNRB, you get different things that are differentially spliced. However, what is more interesting is that we might be keying on one pathway that is impacted more when you start messing with these beyond p53. That might not be tissue specific, which is kind of cool, unexpected.
That was exciting, thank you. Do you know if the mutations that you find in patients, are they dominant negative or is it just haplo-insufficient?
So right now, the idea is that they're haplo-insufficient. The evidence for that, well, it's based on, sequencing patients. So I think only one group has really looked at the SF3B4 mutations themselves.
So in mice, you can't rescue them by adding the wild type protein?
I've never tried.
Okay.
Yeah. That would be interesting.
And is there a single gene that this all boils down to that you can rescue by a splice switching oligo or some such thing?
So we've actually tried the splice switching oligos for the SNRB mutation because that particular mutation is actually a point mutation that alters splicing of another poison exon in that gene itself. It didn't rescue in the cells at least. So we haven't looked at it in the mouse.
Sorry, could you pass that down? Thank you. Hi, thanks for the great talk. So migration is an inherently stressful process, and I know a lot of cells will activate stress pathways. I'm just curious as to whether or not, if you block the migration of the neural crest cells, do the cells survive longer than they would do And is this boiling down to a question of migration being too stressful for cells which aren't capable of activating survival pathways?
That's a really interesting idea. I've never tried that. I can tell you that when we removed P53, what was really surprising, at least when we looked at it really deeply for EF2D2, is that we rescued the survival, but we still had splicing defects. And in fact, we had a higher level of splicing defects. And so I think that if those cells remain in place, they probably would also die because they would have so many things that have gone wrong.
Cool. Thanks very much.
Sure.
Maybe in the sake of time, we'll move on. If any other questions for the break for Loydie? Thank you so much. That's great talk.
Yes, I'm Andrew Davidson. Just recently I set up my lab at the University of Glasgow, and my research interest lies in fundamental tumour biology. More specifically, live imaging tumour cell death within Drosophila in order to investigate how cell death can be paradoxically pro-tumorigenic, meaning it can actually sometimes help a cancer to grow. So I guess from the earliest days of our training, biologists are taught that cell death is inherently anti-tumorigenic, and cancer must evade it if it's to successfully grow and form a tumour. It's one of the hallmarks we all learn about. And certainly when cancerous cells arise in our tissue, they're very effectively eliminated by apoptotic cell death, the best studied form of programmed cell death. However, we know that cancerous cells can escape elimination and grow to form tumours, but surprisingly, the most aggressive tumours commonly exhibit high levels of apoptosis, and this correlates with poor prognosis. Now, beyond a mere correlation, there's good emerging and indeed good historical evidence that apoptosis can be pro-tumorigenic, and a lot of our mechanistic understanding of this comes from work in Drosophila. Okay, so my interest is in the fundamental role of cell death during tumorigenesis, but this, of course, has big implications for how we go about trying to treat cancer in the clinic. After all, the majority of our therapies work by trying to increase tumour cell death to provoke tumour death and then ultimately remission. However, the simultaneous pro-tumorigenic aspects of apoptosis and cell death undercut such efforts, fueling tumour regrowth and ultimately contributing to recurrence. So if tumours can be full of dead cells, then one of the things I think about all the time, well, what happens to all these dying cells? And normally during development and acute injury, it's our macrophages that have this critical role in clearing our tissues of cellular debris. And so my specific research interest lies in tumours, cell death, macrophages, and the relationship between all these and how they might contribute to cancer progression. Okay, so my background is in studying cell death and development and acute injury, and during my postdoc, I collaborated with the Ravichandran lab and a student therein called Mike Raymond to develop a novel genetically encoded probe of apoptosis and its clearance, which we called Charon. So this had two components. It had a caspase-activated GFP, and of course caspase activation is a defining feature of apoptosis, but it also had this novel acid-activated RFP, which is only fluoresces at low pH. And the rationale behind this probe was if a cell dies specifically by apoptosis, it's going to turn green. And then if that corpse is engulfed by a macrophage and internalized, it's going to fluoresce red within the acidic environment of the phagal lysosome, allowing us to visualize the key steps of this cell death and clearance. And my role in this collaboration was to get this into Drosophila so we could perform some in vivo experiments. And my background is in using the embryo and pupal life stages, which allows us to combine the power of Drosophila's genetics with the excellent in vivo imaging you can do in these translucent and immobile life stages, as we saw from Helen's award talk last night. Okay, so here is the Charon embryo, and here you can see the developing cell death within the Drosophila embryo, and the different colours indicate the different stages of this developmental clearance. But of course, this is still the real advantage of this is our ability to do in vivo time-lapse imaging within the embryo. And that's what this is here, and we've also co-labeled the macrophages independently with GFP, and they're undergoing this developmental dispersal, and during which they have this critical role in clearing this developmental apoptosis. And we can now visualize and quantify this through counting the number of Charon red positive corpses inside these macrophages. But if we look at one particularly good example, inside this circle, we're going to see an apoptotic cell light up with our char on green. And this macrophage from the top, which is also labeled in green, is going to pounce on this corpse. And now we can track this corpse inside this macrophage through the activation of the char on red. And we can now track it as it moves around inside the macrophage, inside the embryo. And so we use Charon to quantify this developmental clearance in high detail. But to cut a long story short, one of the big conclusions we found was that macrophage, or the phagocytic capacity of macrophages, can be overwhelmed with too much cell death. Now, normally this isn't a problem, but it got me thinking about pathological settings where there's chronically elevated levels of cell death, as seen in many diseases, including cancer, and how this impaired clearance might exacerbate disease. Okay, but before coming to cancer, with my background in development, I had to develop a tumour model. And before I get to that, I need to introduce just one other biosensor. So this was the Charon biosensor we developed, but the rest of the talk is going to make use of this unpublished variant that we call CHAR-OFF, right? And compared to CHAR-ON, the GFP is constitutively active, so living cells are green, and then if they die specifically by apoptosis, they turn red. And when I was thinking about developing a tumour model, I had the idea that if I could confine CHAR-OFF's expression to a transformed tissue using Drosophila's genetics, I could now live image the sort of growing and dying parts of a tumour in real time. Of course, the embryo is too short-lived to support tumour genesis, so the other big change I had to make was switching to Drosophila pupae, and the reasons for this are largely that the pupae is, like the embryo, great for imaging. It's translucent, it's immobile. The pupal wing, which we're going to focus on, is a simple tissue. It's essentially an epithelial tube full of macrophages. But probably the most important reason is it's derived from the Drosophila wing disc, and without getting sucked into Drosophila developmental biology, this was the classic model of Drosophila tumour genesis, albeit limited to ex vivo and fixed tissue. And so I guess I reasoned that I could take that knowledge and tools and apply it to a setting where I could perform live imaging. Okay, so here are said tools, and now we can use this to express CHAR-OFF in a range of different regions throughout the pupal wing and image it live. And how this works, so if we take one of these models where we express CHAR-OFF in half the wing, we can now live image the development of this wing in green and the accumulating cell death therein in red. And Jane, who's worked on this, has very carefully quantified the accumulation of apoptosis therein. Okay, so that's normal developmental apoptosis. What about a tumour setting? Okay, so again, to highlight what we want to do here, we want to combine CHAR-OFF expression with oncogene expression, etc., and then live image the tumour outgrowth in green and the apoptosis therein in red. Okay, and I don't have time to really go into our tumour model, but if you look at that last panel on the furthest away from me, you can now get this idea of this tumour overgrowth within the wing and accumulating apoptosis therein, if you can see that with this light. Okay, but moving on to the imaging of the macrophages. So here, looking at a slightly different model, here we just express the CHAR-OFF in this stripe running down the middle. And we like this model because it gives a great view of the macrophages within the wing, which we can independently label with GFP. And this allows us to do this long-term intensive imaging. Okay, so here we've flipped it on 90 degrees to allow it to fit us in the screen. And now you can see these macrophages, these dynamic macrophages, interacting with this CHAR-OFF labeled tissue. And if we look at one particularly good example, you can see how this macrophage snakes its way towards this CHAR-OFF red labeled cell. It envelops it, and when it moves away, you can now distinctly see that CHAR-OFF red labeled corpse inside it. Now, of course, this is developmental apoptosis and its clearance, but hopefully you can see we can now combine these two models to give us this opportunity to live image tumour cell death and how it's cleared by macrophages. Okay, I want to stop there and thank everyone in Glasgow that's helped set up the lab, particularly Jane, the PhD student, who really helped drive the generation of this model. The funders for helping me set up the lab. We are also actively recruiting a postdoc, so anyone interested, please do get in touch. And then I also got the great news today from the BSCB that they're going to sponsor our one-day meeting in Glasgow in cell death. So anyone that's interested in cell death and or pirate-themed biology, please join us in Glasgow on October the 6th. Thank you very much, and I'll take questions.
Hey, nice talk. I was wondering whether I have seen in one of your plots when you were measuring over time char off that you have an increase in the accumulation of apoptotic cells.
Yes.
Do you think you have this increase because there is an increase in the cell death rate or because of your technique?
Yes, it's a good question. So in this case, yes, what we're doing, what I've not had time is that we can temporarily control the expression of char-off, and we only turn it on shortly before imaging. And then I think that accumulation is in part a steady rate of apoptosis over time, right? So the other important thing that we know is that the vast majority of that cell death is already inside macrophages, and we know that through using our char-on probe, which I wasn't able to show. So you're right, I think there's this increase of cell death from when we start labeling due to our technique, but it's all very quickly being accumulating in the macrophages, and very little of that cell death is ever within the wing. It's so rapidly cleared.
And do you see it stabilized afterwards?
Sorry, could you say that again?
Do you see it stable? It plateaus?
Yes, so we haven't been able to follow that. It starts getting difficult after the 24 hours because the wing starts folding up, and all that cell death inside those macrophages gets swept into the veins, so it becomes very hard to quantify. What would be a really good way of doing it, an experiment we should do, is to do a sort of pulse of this at different times, and then we can sort of better tease apart the sort of things you're asking. But that would be very easy to do with our model. And our ability to temporally control its expression.
Thank you.
And I think for the interest of time, we will move on to our next speaker, but thank Andrew again for a great talk.
Very good afternoon, and I'd like to start by thanking the organizers and the Company of Biologists for this opportunity to present our work to you today. So my lab is interested in, like so many other people, understanding how cells make decisions and stem cells make lineage, fate, and choices. And we study this right from the beginning of development and trying to understand what happens in aging and in various diseases, because when this process is disrupted, there is a loss of homeostasis and there is dysfunction and disease. And while we study this in multiple contexts and multiple model systems, today I'd like to talk about the blood system and the process of hematopoiesis, or the development of the blood stem cell or the hematopoietic stem cell, which undergoes a series of divisions to give rise to multi-stages of progenitors, which give the classical lymphoid or myeloid lineage of blood cells as we know them in the bone marrow. So making functional blood stem cells, which will repopulate the entire animal in vitro, is currently a major challenge. And this is essential to make because of the paucity of blood donors in many cases, or for the treatment and by gene editing of various disorders. So one of our goals is to make these cells from pluripotent stem cells, which are the earliest cells that form the embryo, and to try and understand the biology of what happens in making these decisions, going from the pluripotent stem cell to the hematopoietic stem cell that will repopulate. And we are also interested in understanding this to find ways in which we can assess the repopulation potential of the hematopoietic stem cells, for example, for studies like transplantation, because currently the efficiency of transplantation is very unpredictable when one is using bone marrow stem cells. One of the sort of major reasons why the efficiency of stem cells repopulating is unpredictable is thought to be because of stem cell heterogeneity. Excuse me. And earlier we have shown that in human pluripotent stem cells, the activity of the mitochondria or the metabolic substates result in the stem cells being able to self-renew or differentiate depending on whether they are glycolysis primed or OXPHOS primed. And this can be a way of modulating or tailoring developmental diversity. But this is something that happens, in an artificial system, in a culture dish. But we also showed that the same is true if we look at an in vivo system, such as the blood system in Drosophila, where you can look at the entire hematopoietic system, in a single animal in one experiment at one time. And there we could show that it's the mitochondrial energy metabolism and fission-fusion dynamics that can actually determine what the cells will do and what lineage the progenitors will take. So they give rise to this heterogeneity at the level of the progenitors. Coming to the vertebrate system and the mouse bone marrow, what is shown is that the hematopoietic stem cells are, heterogeneous by various parameters. Their state depends on the genetic and epigenetic factors, cell intrinsic and cell extrinsic factors, but mainly mitochondrial homeostasis is very important, such that you have two states of the blood stem cell. It could be a quiescent state with, fragmented, immature mitochondria and low levels of ROS, or an activated state with mature mitochondria, high levels of ROS. And depending on this state, their repopulating ability varies. So the metabolic parameters of the HSCs are, governing their quiescence, proliferation, their differentiation, and, also these properties change with age, and pharmacologically one can modulate these to kind of change or reverse the phenotype. But, when one is looking at mitochondria or anything to do with metabolism, it's subject to be influenced by so many other things and so many factors. And in the case of the bone marrow, one samples a small number of cells and extrapolates to the entire organism. So just like we did in Drosophila, we wanted to have a way to look at the entire system and also to try and understand what the ontogeny of this mitochondrial heterogeneity that controls HSCs might be, so that possibly one can control these. Now, the other problem in the vertebrate system is that there are shifting sites of hematopoiesis. In the adult alone, there is the bone marrow, the spleen, liver, kidney, and so on. And there are reports showing that not just in the adult, but even in the embryonic hematopoiesis, there are many factors such as upregulation of oxidative phosphorylation or the change between glycolysis and OXPHOS that can give rise to sort of many changes in the composition or the heterogeneity of the blood progenitors. So if you look earlier on, the bone marrow stem cells come from the fetal liver, which in turn come from the definitive hematopoietic process in mouse and in vertebrates, which is from the aortogonad mesonephros early on in development. So in this situation, there is a hemogenic endothelium that gives rise to an early, stem cell type, the sort of immature hematopoietic stem cell, which then gives rise to a mature hematopoietic stem cell. So we wanted to know, where this mitochondrial heterogeneity that is seen in the adult blood stem cells, where does it arise, and does it arise early on, and what factors influence this? And so to do that, Aishwarya Prakash, an extremely talented graduate student, decided to look at this process called endothelial to hematopoietic transition, where what you see there is the aortogonad mesonephros region sectioned from a mouse embryo, and the green shows you the endothelial lining at the floor of the dorsal aorta. And what you can see on the left is some of the cells in red are the hematopoietic cells that are budding off from the endothelium, and then these go on to completely bud off and form these mature hematopoietic stem cells in this process called EHT. So what Aishwarya did was initially in the sort of sections of the embryo, without isolating the cells out of context, because this is going to affect the mitochondria, looked very painstakingly and in depth at the nature of the mitochondria, mitochondrial morphology, activity, and so on in the green cells, in the cells lining the endothelium, as well as in the c-KIT positive hematopoietic hematopoietic stem cells. And what she saw was that in the endothelial cells, while the mitochondria are elongated and mature, as the hematopoietic stem cells arise, the mitochondria get fragmented, and their characteristics and morphology changes. And to summarize a lot of work and a lot of characterization that she did, in the endothelial cells, like I said, the mitochondria are more mature. As the hematopoietic stem cells emerge, the left part of the schematic, there is an increase in several mitochondrial parameters, such as the membrane potential, the mass, activity, ROS, calcium, and so on. And as the cells mature from the type 1 to the type 2, or the mature hematopoietic stem cells, actually there's a decrease in all these characteristics, with one exception being the mitochondrial membrane potential that continues to sort of remain high. So to see whether this actually affects what the cells are going to become, she used CCCP to reduce the potential, or mitoQ to increase the membrane potential, and did this either ex vivo or in utero. And what she found is that the early stages of stem cell emergence are not really affected, but it's only the maturation of the mitochondria of the hematopoietic stem cells that is affected by changing or by reducing the mitochondrial membrane potential. You end up increasing the output of mature hematopoietic stem cells. And to test this further, she did an RNA-seq and found that it's actually the Wnt signaling that is upregulated in response to reduced mitochondrial activity, and you could inhibit this Wnt signaling and rescue the phenotype. So to summarize, so far, the HSC emergence results in huge remodeling in different stages of the mitochondrial activity, and this is regulated by the activation of Wnt signaling. So how does this affect the function of the mitochondria? So to do that, we looked closely at the mature emerging HSCs, and what was very striking is when you look at just the FACS plots, what you can see is as the endothelial cells become the emerging HSCs and then the mature HSCs, there's a clear shift in the mitochondrial membrane potential, such that in the mature HSCs you actually have distinct subpopulations which are either high or low in the mitochondrial membrane potential. And to again test whether this matters and what it means, she did a lot of in vitro and in vivo analysis. But to cut a long story short, what she could show that if you transplant these two populations separately, the low MMP and the high MMP populations, you get differences in the way the sort of transplanted cells repopulate the irradiated mice, such that the cells which are higher in their mitochondrial membrane potential seem to be more quiescent, and they are better able to populate the irradiated mice. So again, to try and understand what this means in terms of the signaling, what the mitochondrial potential is doing to the signaling, she resorted to looking at a single cell RNA-seq and did a lot of detailed characterization of the signaling and the pathways. But what was very striking is that when you compare the cells, the hematopoietic stem cells that have low versus high mitochondrial membrane potential, what you see is that they segregate very nicely into different kinds of HSC subtypes and gene expression clusters, such that the low MMP cells are more myeloid primed, whereas the high potential cells are more lymphoid primed. And she went on to then look at what are the signaling pathways, and it turns out it's that PI3K signaling that seems to be perturbed, such that if it's reduced when you have increased membrane potential, and you can actually either increase or reduce PI3K signaling and reverse the effect. So to summarize, what we could show that between emergence of the hematopoietic stem cells and their maturation, there are myeloid and lymphoid lineages segregate right at this stage, at the very first stage of hematopoietic emergence. Based on the mitochondrial membrane potential, you can actually decide which lineage to make. And going forward, of course, we wanted to know how this really is going to affect adult hematopoiesis, and what we did was let the mice where we had manipulated the membrane potential in the embryo go reach adulthood. And indeed, if you reduce the embryonic membrane potential of the HSCs, where you see expansion of the hematopoietic stem cells, it actually results in expansion of the hematopoietic population, even in the adult bone marrow. So this is, of course, pharmacological modulation. We've also done genetic modulation using a mouse model, which is a mitochondrial protein, which, when we have a mutant, we have a knockout of it, it leads to bone marrow expansion and myeloproliferation. And even there, the knockout HSCs have low membrane potential. So these also age prematurely, and what we've shown earlier is that we can, put back expression of the protein and to an extent rescue this phenotype. So what we want to do now is we've made a reporter mouse for this gene usrage that we used earlier, and this also very nicely, the reporter segregates into the heterogeneous expression pattern. And what we are now trying to do is to sort of, use this reporter to screen for modulators of the mitochondrial activity, either high or low activity. And we have a few hits that we aim to pursue to see if we can modulate these during embryogenesis. We're also trying to use human embryonic stem cells, where if we differentiate them with the endothelial to hematopoietic transition protocol, we do see segregation of the high and low pools. And our early experiments suggest that the way the high and low pools behave when we try to differentiate them through a 3D protocol to try and generate early embryonic structures, the low MMP low expressing cells don't seem to be very good at aggregating to form these embryo-like structures. And what we're doing is we're taking now the gastroloids model, which is the first step of forming the mesoderm, from which we will get the hematopoietic system later. And this is now very well standardized, and what we aim to do is to actually make the tools and reagents to track this and to try and modulate, because development occurs in 3D and metabolic activity also determines on the 3D organization of the embryo. So to summarize what I've told you is that in development, using the hematopoietic stem cells as an example, we show that the heterogeneity can be because of the mitochondrial activity, and this can be used to tune the development so that you go from a calm state to a stress state. But importantly, this is going to vary from one tissue to the other, and one needs to keep this in mind while looking at the context in which a particular sort of mitochondrial activity may modulate and what the outcome may be. So with that, I'd like to stop and thank all the people involved. Mainly I've talked about the work of one graduate student, Aishwarya, but there are many other wonderful students and collaborators whose work I haven't been able to talk about. And I'll be happy to take questions. Thank you.
No, go on, we'll pass it back.
Thank you, Maneesha. I was just wondering, what determines, first of all, whether the membrane potential is low or high, and the distribution of it in nature? And if you were, since you're isolating the stem cells of different membrane potentials, if you were to, I guess, culture them in vitro, do you regenerate the normal in vivo distribution of membrane potentials? So I'm asking, is this heterogeneity intrinsic? How does it arise?
Yeah, so we're not sure how it arises, but the whole process is triggered off by blood flow and then RUNX1 getting expressed, so that you have the process of endothelial cells becoming hematopoietic cells. If we know, like I showed you earlier, that maybe some of the signaling, for example, wind signaling, may be responsible for sort of triggering off. But I expect there is not going to be any one trigger alone for this metabolic activity. And sort of coming to the second part of your question, what about culture? So as far as possible, we have avoided doing the culture because a lot of studies from the bone marrow field have actually taken the cells and cultured them and then shown differences, but we wanted to avoid those kind of artifacts. But when we do culture these cells, so we did a lot of in vitro analysis also. And when we do culture these, sort of isolate the cells and culture them, that's when we also first see the phenotypes of whether they can, self-renew or differentiate. And the same holds true in vitro and in vivo.
And do they regenerate with distribution?
We did not look for that, just because there's always the caveat of we don't know what happens in culture, because a lot of the culture media use modulators of mitochondrial activity and with them. Okay. So this one can do in the premise. So
just a question trying to incorporate if actually the increase of mitochondrial activity, what is the trigger? Is that ROS? And perhaps, what is the trigger basically? Is that the consequences of the increase of mitochondrial activity? Is that really the ROS productions that could change something in the state of differentiation of the cells, or actually any underlying metabolic remodeling associated with an increase of mitochondrial respirations that really influence the phenotype of the cells?
So, of course, ROS is well known to be one of the sort of outputs of increased mitochondrial activity, but there is increasing evidence showing that there is no sort of definite one-on-one correlation. So higher ROS doesn't always mean increased metabolic activity and so on. So we don't know what the direct connection is, but we have shown with the bone marrow hematopoietic cells that increased activity leads to also connects to the pyruvate or the glutamate levels and parts of the metabolic cycle being modulated. But specifically what happens in the emergence is not something that we have looked at yet. It's sort of challenging to look at it unless, again, you isolate the cells and culture them. Because people have even done things like seed-host analysis on the emerging sort of stem cells, but that's again pulling embryos, isolating and purifying cells. So that gets technically a little challenging to produce.
I think in the interest of time, we'll let the last question come in, which is the coffee break in between, and move on to our flash talks after. Thank you, Maneesha.
So one of the projects we have in our lab is to understand how human hindbrain develops and what goes wrong in Down syndrome. So Down syndrome, as you probably know, it's associated with an extra copy of chromosome 21, also called trisomy 21. And it coincides with intellectual disability and between many other phenotypes, but also to small brain phenotype, being the hindbrain and particularly the cerebellum, the most affected brain region. Okay, so in the lab, we are studying human brain development, but our postdoc and the work I'm showing today from Teresa Silva, she's using hindbrain spinal organoids to understand the development and the progression of the disease in Down syndrome. And what she's observing, if you compare the control with the trisomy 21 derived organoids, is that we lose the apical surfaces in Down syndrome. And at the apical surface, as we have here exemplifying this terrible image, is that the progenitor populations, that's where they live, so the apical progenitors. And the size of this pool, it's known to correlate with the final size of the brain. Now, she also observed that ECM components that are localized generally in the epithelium at the basal surface, she found that they were altered in Down syndrome. And when she adds ECM to the media, when she's culturing trisomy 21 derived organoids, she rescues the phenotype. So again, if you know about ECM and cell polarization, please find me in my poster 39. Thank you.
up, we have Jessie Ju from University of Cambridge.
Okay. Hello, everyone. I'm Jessie. I'm a PhD student at Cambridge studying GCN2, a ribosome stress-sensing kinase in the integrated stress response that often gets activated during ribosome stalling with help mediated by adapter GCN1. To study the biology of GCN2 and its non-connection to high blood pressure in the lungs, we performed a chemical screen of over 6,000 compounds for GCN2 activators, yielding 3 lead molecules. Biochemical assays showed mechanisms and kinetics of my compound, where it competes with ATP for binding to GCN2. Cell-based assays similarly showed a reduced Brett signal indicative of my molecule outcompeting a tracer from binding to GCN2. And computational modeling showed predicted docking of my molecule in the GCN2 ATP binding pocket. Interestingly, my lead compound 20 activates GCN2 independently of GCN1, which is in contrast to a non-GCN2 activator, neratinib. We're still working on the mechanism behind this GCN1 independence, but we hypothesize that it could be due to the molecule's small size, fitting very easily into the ATP binding pocket. So in summary, I've identified a GCN2 specific, GCN1 independent activator that can be used to better understand the complex biology of GCN2. Please come visit my poster at 79. Thank you.
Hi, everybody. I have no slides, so you can just look at me or look awkwardly at the floor. We study gestational hyperandrogenism, which is seen in conditions like women who have polycystic ovarian syndrome who get pregnant. The fetus is exposed to slightly higher than normal levels of androgens, and this has been associated in human epidemiological studies and work in sheep with cardiovascular defects after the fetus is born and develops. And we use zebrafish to study this in the lab. So if we add androgens to the water in very precise amounts, concentrations, we can regulate androgen levels in the embryo, and we find that there are cardiovascular defects, which I can tell you more about at poster 61. And these are mediated not by the nuclear androgen receptor, the canonical signaling pathway, but by a G protein coupled receptor, a GPRC6A. So if you're interested in learning more about that, come to poster 61. And if you're interested in being paid to peer review manuscripts, I'm also the editor-in-chief of the journal Biology Open. We're starting a new experiment where we're paying peer reviewers. Come find me anytime at the meeting. Happy to talk to you. Thanks.
Thank you. So the size and complexity of the human neural cortex is what essentially underpins our sentience, but for these reasons it makes us susceptible to conditions like microcephaly. So over 15% of inherited cases are caused by mutations in WDR62, and these mutations essentially affect how the millions of neural progenitor cells divide and differentiate and migrate to expand the neocortex. But interestingly, mutations in purine metabolizing enzymes share strikingly similar defects. What I've been able to show is that WDR62 actually co-localizes with many of these purine enzymes in these punctate bimolecular condensates known as purinosomes. And also it interacts with a number of molecular chaperone proteins, including the one on the top right there called BAG2, which has specific roles in the folding and turnover of these enzymes. Specifically, I showed that when WDR62 is lost, this appears to shift the proteostatic balance from folding to degradation and results in the enhanced breakdown of a particular purine salvage enzyme called HPRT. I show that this in turn disrupts purine homeostasis and actually impairs neurodevelopment in a way that closely mirrors the loss of WDR62. So if you're interested in this novel mechanism of neurometabolism, come along and see my poster at number 63. Thanks very much.